start then, yeah. Hi, good afternoon. Welcome to today's weekly research conference at the Ottawa Heart Institute, also at the University of Ottawa and the Ottawa region. And just a few logistics before we introduce our esteemed speaker today. So make sure that you submit your questions using the Q&A feature on the bottom of your screen. And they will be answered at the end of the presentation, but feel free to enter the questions as the lecture is proceeding. And also we'll be accepting live questions at the end of presentation. You can use the hands up feature and our moderators will be able to unmute you to allow you to ask the questions in person. And but on the other hand, you can use the chat feature if you're experiencing any technical difficulties. And Kelsey and Allison and the members of the team will try to troubleshoot with you and have you join successfully. So today we're absolutely privileged to feature another of our trainee committee's special outstanding guest lecturer. And we have the privilege of having Dr. Michelle James, in fact, interacting with the trainee already. And we really appreciate her taking time out on this occasion. And we welcome Dr. Michelle James, who is an assistant professor in the Department of Radiology and Neurology at Stanford University in beautiful sunny California, as a member of the renowned molecular imaging program. The molecular imaging program started with the late famous Sam Gebeer, who is a real genius in this field. And with other prominent members, including Catherine Ferrara, Dr. Joseph Wu, who worked together very closely on multiple areas, and Dr. Brian Rutt, who's also Canadian, who's joined the Stanford team. Dr. James received her BS in pharmacology and medicinal chemistry at the University of Sydney, where she also earned a PhD in radiochemistry, neuropharmacology, and was awarded the prestigious University Medal. And through her postdoctoral fellowship, and a lot of done at Stanford and now on faculty at Stanford, Dr. James' research focused on the developing and evaluating of novel molecular imaging agents to improve the ways in which you diagnose, treat, and understand a variety of brain diseases. Her lab's special approach is designing of new PET radioligands for visualizing neuroinflammation. Her team discovers and characterizes a number of immune cells and their markers. And then her team will develop specific radioligands to target these markers as a diagnostic tool or as therapeutic targets. These are applicable potentially to Alzheimer's disease, multiple sclerosis, Parkinson's disease, and stroke, with the goal of learning about these innate and adaptive immune cells in the early stages of disease formation and the contribution to the disease progression. A special tribute to Dr. James' research is really the ability to translate these novel molecular tools to the bedside to track disease progression and guide therapeutic selection for individual patients as potential also surrogate endpoints in clinical trials. These are usually Herculean challenges, but Dr. James has already developed multiple patented radioligands for imaging these neurological conditions, 3 of which are currently being evaluated in humans in various stages of clinical trials. And for these amazing accomplishments, we really look forward to hearing the journey that she has undertaken behind these discoveries. So please welcome together Dr. Michelle James, who will discuss with us today the topic of molecular imaging of immune cells in the whole body and brain, a critical strategy to improve the treatment of neurological diseases. Thank you.
Thank you so much for that introduction, and to the trainee council for inviting me. I was just saying to them this morning, I'm sad that I can't be there with you I really enjoyed visiting Ottawa the last time I was there. I am not sad to be missing the cold weather though, because I remember when I was there last, Adam Schuhandler was making fun of me because I really couldn't handle the cold, and it wasn't even that cold. I think it was October, but I was crying because I was so cold. But anyway, I'm from Australia originally, as you could tell from where I did my education, and so I'm used to warm weather. And so we have California's been a nice transition for me. That's where I can actually survive most of the year. But I would definitely come visit you in summer. So if you want me to come up there, that would be a good time. So let me get this started. Okay, so Peter's already given a great introduction about what my lab is focused on, so I won't spend too much time, but just to say that we're really interested in trying to understand the role of the immune system and sort of different aspects of the immune response in the development and progression of brain diseases. So my lab really does everything from discovering different biomarkers that would be very specific for different cells or processes that are important for the immune response, to then we validate those biomarkers using human tissue, rodent models, and so forth. And then once we're happy with the specificity of a biomarker, we go about the big undertaking of making a PET tracer to detect these biomarkers in living subjects. So we'll do all the chemistry. And at that point, we're also interested, of course, in validating the imaging agent, in using different rodent models of diseases like MS and Alzheimer's and stroke, as you'll see in some of the slides I'll be sharing. But we don't want to spend forever in animal models. We want to really try to get to the clinic as quickly as possible. And that's one of the benefits of PET and radiochemistry, is that you can really quite rapidly get your molecule into people if it looks good in the preclinical development. It's not too expensive or time consuming if it's a small molecule. If it's a biologic, it's a different story, but we can talk about that. But in any case, I think it's really powerful to be able to get these molecules into people so we can study disease relevant processes in the clinic. Okay, so my talk today is divided into three main parts, and I'll start with just a little bit of background about why I'm so interested in imaging the immune system and different immune cells. And as part of that background, I'll introduce some of the key imaging targets that myself and others around the world have been exploring and developing PET tracers for, so that they can visualize different parts of the immune response. And then as part of that, I'll very briefly talk about how my lab has been targeting one of these imaging biomarkers, the most widely investigated imaging biomarker for neuroinflammation called TSPO, to monitor neuroinflammation in chronic fatigue syndrome patients. And that's some of our more recent, I think, very exciting data where we're really looking at what's happening in a very mysterious illness. And then I'll finish by sharing a couple of newer strategies that my lab has been working on to track immune cells in stroke, Parkinson's, sepsis, and MS. One of the strategies is really probing myeloid cells, which you can see here is the middle topic. And the last topic is talking more about how we are targeting adaptive immune cells, and in particular B cells in MS. So let's get into it. Okay, so what is neuroinflammation? Why do I care about it? Well, it is any inflammatory process that is occurring in the central nervous system. It typically involves microglia, which are your brain's resident macrophages and immune effector cells, the first cells to jump into action whenever there's some kind of injury or infection. Also, your astrocytes are involved. Oftentimes, peripheral immune cells are recruited into the central nervous system to help out. There's a lot of production of cytokines, chemokines, and really in its acute form, neuroinflammation is beneficial. It's there for a reason. We have an immune response and inflammation there to help heal and to clear away damaged tissues, to mitigate pathogens. So it's a good thing. But the problem is when inflammation becomes unregulated, when it persists and just it's not resolved, that's when it can become quite problematic. And this is when it leads to cell dysfunction, neuronal death, and things that you see in neurodegenerative diseases. So that's what I'm interested in understanding more about. Why does it become sort of maladaptive and chronic, and how can we reverse that or modulate it? And so neuroinflammation, like why we actually really care about it, is it's actually a key underlying feature of pretty much every brain disease. And I know most of you here are not working on brain diseases, but you're working on, I'm speaking to in the Heart Institute meeting right now. So you're doing a lot of things to do with atherosclerosis or MI and all sorts of things, diabetes. But inflammation as a whole is involved in pretty much every disease. So I think it's important that we try to understand more about the immune response. And that's where I think that PET imaging and imaging in general, and I'm a little bit biased towards PET because of its translational capabilities and sensitivity and ability to look at molecular changes quite quickly, is that the power is that we can really start to understand some of these processes by developing specific agents, so we can look at them in vivo. And in particular, the whole field of a lot of brain diseases, and in particular Alzheimer's, for instance, has been flipped on its head more recently, whereby GWAS data, sort of this genome-wide association studies, has shown that the main genetic risk factor that increases your likelihood of getting Alzheimer's is really to do with your immune response and your innate immune cells becoming dysfunctional, sort of losing their proper functioning abilities. And so again, I think that this highlights that not only should we be developing therapies that modulate the immune response, which is what a lot of pharma is doing right now to help to treat Alzheimer's, but we need to have imaging to complement that so that we can understand what these cells are doing, so that we can evaluate whether the therapies are working or who would be best for these therapies. And I think we can really apply a lot of the tools that I'll talk about today. I think you could apply them to different diseases that you're probably more focused on in your institute. So I'd love to talk more about that. But in terms of neuroinflammation, cellular and molecular imaging biomarkers that people have been looking at, I'll show you a few cell types, and in red are the markers that I think are the most common ones that people have been looking at to make traces for. So microglia, we've got a list here, TSPO, definitely the common, but then newer targets like CSF1R, P2Y12, and some others, astrocytes, people have been looking at. We'll see TSPO again because it's not just specific to microglia. It's also expressed on astrocytes when they become activated, but to a lesser extent. GSK3, there's been a number of groups, Neil Bastev and Peter Scott's group have been working on developing traces for that target. I think it's quite interesting, although it does appear to be expressed by neurons and endothelial cells as well as astrocytes. So you have to be keeping in mind what these targets, or at least imaging these targets, will reflect in terms of the final output of a PET image. What is that really telling you? What are you looking at? I think we want to be as specific as possible, of course. Peripheral myeloid cells, which are my personal favorite cells. I think they're just fascinating. Your macrophages, neutrophils, monocytes, and how they can actually get into the central nervous system. They really aren't used to being there. So I think that they have more of a propensity to cause trouble, but they're just not used to being in that environment. And so I think developing tools where we can sort of pass out the peripheral myeloid cells from your brain's resident myeloid cells, the microglia, is particularly helpful. And my lab has been working very actively in that area. And I'll share a little bit about that through this talk. And then you've got your adaptive immune cells, so your B cells and your T cells. And Anna Wu's group down at City of Hope, she used to be at UCLA, has done a lot in that space, looking at CD20, but also in the T cell imaging space, CD4, CD8. She started Imaginab and has that CD8 mini body for looking at T cells. And then Sam Gambier's group had developed a really great imaging approach for looking at a T cell activation marker, OX40. So there's a range of different tools and imaging biomarkers you can look at. Also, of course, you can look at cytokine signaling, oxidative stress, which I know Adam Schuhendler's group has done a lot in that area, and it's really contributed in a meaningful way in that space. I think oxidative stress is underlying many diseases and is really intimately associated with inflammation. And so I won't really be talking about those today, but I'll be focusing more on these targets. So a little bit about TSPO in the next slides, and then I'll dive more into TREM1, which allows us just to look at peripheral myeloid cells, and then I'll talk a little bit as well about looking at B cells with CD20 and CD19. So a little bit about this TSPO, which some of you who are in the imaging space would have heard a lot about. And for those of you who have not heard about it so much, it's really this highly hydrophobic 18 kilodalton protein believed to form part of this multimeric protein complex in the outer mitochondrial membrane. It's important. It has an important role in the synthesis of neurosteroids by promoting the transport of cholesterol from the outer to the inner mitochondrial membrane, which is really the rate limiting step in neurosteroidogenesis. But the interesting thing with TSPO is that it's present at very quite low levels in the healthy central nervous system, but then it becomes overexpressed in different neurodegenerative and inflammatory diseases. So actually people have used it as an imaging biomarker for various heart-related diseases. Even though it is kind of highly expressed in the myocardium itself, people have been using it as a biomarker of inflammation in those diseases as well. So it really does seem to be a marker of innate immune activation or glial activation, because it is predominantly upregulated on microglia, but also on macrophages in the periphery, and on astrocytes to different extents depending on the disease. So it's this active, a marker of active disease or inflammation, I would say. So there's been actually tons and tons and tons of molecules that have been made for this target in different classes, and different PET traces that have been made for this target. And this is just a few of the most common ones, or the ones that are being explored in both rodent models and in the clinical setting. And these are just some examples about how it's been used. I won't talk too much about it, but suffice it to say, TSPO PET imaging agents have been used to look at ALS, multiple sclerosis, Alzheimer's. Here's an example of looking at lower back pain by Marco Logio's group at MGH, where you can see there's a lot more signal in the brain where there is low back pain. So that's a really important finding, mainly in the thalamus actually is where they were seeing increased uptake. And most recently, they were looking at fibromyalgia as well, which I think is really an important disease to try to understand better. And then this image really scares me a lot, which is looking at NFL players just after one single concussion compared to age and sex match controls. And you can see there's a lot more tracer uptake and binding, which is indicative of inflammation or some kind of active immune response. Is that a good thing or a bad thing? We don't really know yet. For the most part, it's probably not great, or at least in some people, there could be a resolving of that inflammation. The inflammation could be helping to heal, whereas other people, maybe it gets into sort of a spiral of toxic inflammation over time that could lead to cognitive decline. The good thing is that we can now track people and sort of try to understand which trajectory they're going down in terms of cognitive decline or susceptibility for cognitive decline. And the thought is that we would hopefully, in an ideal world, be able to catch whether somebody was going down a more toxic or maladaptive route, and that we could intervene with some kind of immune modulator. And so that's just showing you a little bit about how TSPO PET has been used by different groups. I've been increasingly interested in trying to use it to understand more mysterious illnesses of the brain and body. Most recently looking at chronic fatigue syndrome. And the objective here was just to see, can we detect neuroinflammation in chronic fatigue syndrome, which is also called myalgic encephalomyelitis. So we call it MECFS. And so we were looking at moderate to severe patients as per their multi-dimensional fatigue inventory score. They had to have a score between 76 and 100. We had an age range that we're sticking to, and ideally we want to look at 10 and 10 subjects. We're about halfway through that at the moment, so I'll show you some of our first results. These are the scanning parameters. So we're doing PET MRI. We're lucky at Stanford to have 3 actual scanners that allow us to do PET imaging with MRI simultaneously. So these are sort of the sequences that we're using. We're able to look at a lot of different MRI data at the same time as collecting our dynamic PET scan. And this is the molecule that we were using, which is actually something I developed in my PhD many years ago. And it's nice to be able to use that in people now at Stanford. And this is just a very simple molecule. It's a pyrazolapyrimidine, just doing a simple O-alkylation, one-step synthesis here to get our labeled product. And just a little bit more about chronic fatigue syndrome for those of you who don't know too much about it. It's really an awful condition that it's quite mysterious, and there's quite a lot of stigma around it. And it's characterized by this chronic, profound, disabling, and unexplained fatigue. You have unrefreshing sleep. So you could sleep all day, all night, but still feel absolutely exhausted. And it strikes, people that, are otherwise very healthy and are doing, maybe quite remarkable things. And I know it happens, at least there are some examples here where I've interacted with families and the patients where the kids have been, really high achievers, doing great things at school, going to Stanford or other schools, doing lots of great things, but then all of a sudden are struck by this fatigue and just not able to do anything anymore, and they're bedridden, and it's, that can go on for years or decades, and maybe they never recover. And it's not just the fatigue, it's also that there's cognitive impairment, so there's brain fog, there's chronic widespread pain and depression. So it's really an awful condition. Preliminary theories posit that there's an initial trigger, potentially a viral trigger, which I'll circle back to how that ties into COVID-19. But yeah, people think that it's a virus that initially triggers maybe some sort of chronic inflammatory cycle that affects many regions of the body, including muscles, joints, lymphatic system, and the brain. Unfortunately, the standard of care techniques to diagnose it, usually like blood tests, structural brain imaging, they usually all come back negative. And so people think that, oh, there's nothing wrong with you, and doctors are, not able to give you a clear diagnosis. And that's really hard on the patients because they know something's wrong. They're not able to live the way that they used to, but yet all the tests are coming back negative. And so, we're very interested in sort of exploring this inflammatory theory around chronic fatigue syndrome and whether there is inflammation there, whether we can see something, and if so, can we treat people? So in any case, with that background, we were recruiting patients and we're putting them in the PET-MR, and they really are troopers because they're light and sound sensitive, but they were getting into an MRI machine, which is very noisy. And they were, hanging in there and to get it done for the name of science and to try to figure out what's happening. So I would be talking to them saying, you don't have to do the whole scan, just let us know how you go. But they wanted to do it. So I'm really impressed with these patients and very impressed by what we're able to do so far. These are some of the first images that we're able to get. This is a female patient with an age-matched control. And you can see even just by sight that there's a lot more uptake in and around the thalamus and the pons, even in the cortex compared to the healthy control. So certainly something's going on here. And this is my grad student, Mackenzie, who's an engineer, really talented scientist who has been helping to analyze this data because we've never looked at TSPO PET data in this patient population. What we need to do is usually look for a reference region so that we can analyze the data correctly. And so she did that. And this is actually just showing some male images, which looked a little bit different to the female images, which is why it's important to have age and sex matched data because TSPO can change according to gender and also age. So very important to do that. But you can see again that there is more uptake again in and around the pons and thalamus for these patients that have chronic fatigue syndrome. And something else that we noticed was below the head, below the brain, was there was a lot of uptake in and around the glands in all the ME/CFS patients. And I would notice these patients holding their necks and sort of saying they're in pain and their glands were swollen. So that was kind of an interesting finding. And I wasn't sure if it was real, But then we looked at everyone that we'd scanned so far, and actually we've got a few more now. And it seemed to be a common thing that there was a significantly more PET tracer signal in and around these parotid glands. And I did a little bit of digging and reading, and it seemed that actually there's some papers out there to show that there are increased numbers of mast cells, which is an innate immune cell, in the saliva and in salivary glands of these patients that is indicative of an active immune response. So I think what we're seeing here is something real. TSPO is expressed on mast cells. So that got me thinking, what else are we missing here? We're looking at the brain, and that's very useful, of course, but we know that this condition and disease really affects the whole body. So why aren't we looking at the whole body? We've injected this PET tracer into someone. We're just looking at the brain. Why can't we scan the whole body? And so I suggested that, as something we try, and everyone kind of laughed and said, there's no way you can do a whole body scan of a carbon-11 PET tracer because the half-life is only 20 minutes. So how are you going to image the brain and the whole body? And I said, well, let's try it, because we have a very sensitive scanner at Stanford. And so I actually volunteered for the first study of looking at whole body PET-MR imaging of TSPO and inflammation. And so that's me in the scanner looking very happy. And I think I was probably the first person to make a PET tracer and then be injected with it. My own tracers, that was pretty funny. But I wanted to go through it myself first to make sure it was okay before asking these patients that are very sick to have to go through all of that, because the scan was going to be longer and you're going to have all these different, weights on you, different body coils. In any case, I did that. These are the first whole body and brain TSPO PET scans using a carbon-11 tracer, and maybe the first full body TSPO PET scan there is for looking at diseases so far, I think. So I was encouraged that we could see, everything we needed to see, and the areas where we know there's high TSPO, like in the myocardium, in the liver, in some of the intestinal tissues, in the spleen. And so I thought this was a good proof of concept. And actually now we have funding to do whole body and brain imaging of chronic fatigue syndrome patients compared to healthy controls, which we're very much looking forward to getting started on once COVID clears up. We can't scan any patients at the moment, unfortunately. But speaking of COVID, there is, of course, a link between chronic fatigue and COVID-19. And actually, over the past 9 months, there's been an increasing number of people who have reported extreme exhaustion and malaise after having the virus, even months and months and months later. They're struggling to get out of bed or to work for more than a few minutes at a time. So it's sort of this brain fog. And Fauci has been warning against post-viral syndrome, similar to what happened during the SARS outbreak in 2002. And really, as the pandemic continues, we're learning that many people who experience COVID-19 endure these long-term health consequences called post-viral syndrome. And so, really these symptoms that have brain fog and fatigue, maybe related to cytokines that can cross the blood-brain barrier and affect the brain. It's very likely that there's inflammation going on. And we just don't know yet what's happening. And all the experts are saying, well, to assess long-term consequences of something like this, the only thing you need is time. And that's hard on the people that are going through it. I think time is what we need, but also I think imaging techniques to look at what's happening in the brain in real time. So we're going to be starting some studies soon looking at inflammation in the brain of COVID-19 patients. And I think there needs to be a lot of work in that area to better understand what's going on so that maybe we can curb things and hopefully modulate the immune response to be more beneficial with some of these newer drugs that are coming out, and also drugs that are already being used for that purpose for other diseases like MS. Okay, so just to wrap up the TSPO PET side of things, I think that my takeaways for you for thinking about TSPO PET is that it's a pretty good biomarker. It's not the best for sure, but its strengths are that it's really the most widely used and evaluated PET strategy for looking at neuroinflammation and inflammation in general in patients. You can use it in animal models as well, of course. It's given us the unprecedented opportunity to track glial activation and innate immune activation in different diseases. We just haven't been able to do that before. So it's a good first step. But there are limitations, and the list is long. And that's why my lab is focused on really coming up with better biomarkers for neuroinflammation. But some of the limitations are sensitivity is kind of low. The upregulation that occurs in these diseases is pretty small. So if you want to look at subtle changes, your ability to do that is not fantastic. And it's not only expressed on microglia, as I said, also on endothelial cells, astrocytes, even platelets. So you have kind of a messy signal that's confounding our ability to look at one cellular population. So we need to be careful in how we interpret the images. And another thing that I think people don't always think about is that with biomarkers for PET or anything really for imaging, it has moderate to high expression throughout the whole body. So if you're interested in looking at inflammation in a part of the body, for instance, in the heart, in this case, it might be a little bit tricky, even though people do it, but there is already high expression in that tissue. So there might be a better biomarker for what you're looking at that is not TSPO. And I'd always think about that ahead of time. Also, it doesn't provide clear functional information on the phenotypes of these cells and what they're doing. So I think that's also an important question to ask yourself. And that brings me to the next part of the talk, which is what does make an ideal imaging biomarker of neuroinflammation, or anything for that matter. This list could be used for any biomarker really. It's just like an ideal wish list. But this is looking at a bunch of the different targets in the brain. And this is from a review that Purva Jain in my lab published with a bunch of the other students in my lab. But this is kind of our wish list, is that there's optimal signal to noise ratio, so low basal expression in both the CNS and the periphery. So it's better if it's just not there normally and then pops up whenever the disease or a process of interest is occurring. Or, you know, the other way around, maybe that it's high, but then it goes down. But there needs to be a good signal to background so you can look at what's happening with high sensitivity. There needs to, as I said, be a sufficient change in target, and that needs to be correlated with disease severity, ideally. It's nice if there's an adequate concentration of targets, so it's important to think about your Bmax. Of course, you're not always going to have high Bmax for your target of interest. For instance, alpha-synuclein in Parkinson's, pretty low Bmax, and it's diffuse, low levels, so it's hard. It's been a really hard target for people to make PET traces for. You need to then make traces that have super duper high affinity in order for you to be able to see this target. But definitely it's good to have an understanding of what your Bmax is, so your maximum number of available binding sites in your tissue of interest is what the Bmax is. You need to have a high specificity, I think, for a given cell or process. It doesn't have to be just a cell type, it's certainly a process that you're looking at. You don't want it to be contaminated by other processes. And I think it's important to have functional relevance specific to a cellular activation state. So for inflammation, I think it's nice if you have a biomarker that tells you when, for instance, microglia are in a pro-inflammatory state or an anti-inflammatory state or in a more homeostasis supporting state, so that you can really know, to be able to draw more conclusions from what you're looking at. And these are the students that have worked on this really cool review that if you're interested, check it out. And then just to touch upon a couple of emerging biomarkers for microglia and myeloid cells that have come about more recently, there's the CSF1R. A number of groups have been working on this, the group at Hopkins, Marty Pompa's group and Horty, as well as Peter Scott's group have been working on this and others. It is selectively expressed on pro-inflammatory microglia and peripheral myeloid cells. So that's cool. I think that's great. It's definitely a step in the right direction because it's more specific for a cell type, just myeloid cells. Obviously, it can't tell you the difference between the microglia and the peripheral myeloid cells coming into the brain, but it's still a very good step. A handful of traces have been looked at pending clinical translation. And here's just an example of this tracer being evaluated in autoradiography of Alzheimer's brain tissue, just showing high uptake in the AD tissue compared to blocked tissues and the controls. But certainly there's ways to go there to understand how useful this imaging target and traces will be. Then there is the P2Y12R target. It's selectively expressed on more sort of homeostatic or anti-inflammatory microglia, and it's only on microglia in the central nervous system. It's not on peripheral myeloid cells, so that's nice. The traces that have been made, actually by Windhorst's group in the Netherlands, unfortunately have not been able to cross or be retained in the brain. So that's a problem. We haven't been able to evaluate them for imaging neuroinflammation yet. But my group's actually working on a new generation of a tracer, my grad student, Isaac Jackson, and hopefully we'll be testing that soon. We'll have something to share with you. I'm crossing my fingers that it'll cross the blood-brain barrier. We're sort of using some novel ways of predicting that, developed some new sort of machine learning algorithms and things like that to predict whether something will be CNS penetrant. So what I've been focusing on and what I think is a really promising target or biomarker of inflammation in general, not just neuroinflammation, but if you're interested in other inflammatory based diseases, TREM1 is something to look at. This is the triggering receptor expressed on myeloid cells type 1. It's only expressed on myeloid cells, as the name suggests, and it's only there when there's more maladaptive or toxic inflammation occurring. It's linked to both acute, so sepsis, stroke, pneumonia, and chronic inflammatory disorders, so rheumatoid arthritis, inflammatory bowel disease, and it really does seem to be a great biomarker of innate immune activation. We made the first reported PET tracer for this target. There were no small molecules, and I'm a small molecule radiochemist, so I was a little bit nervous and skeptical to label an antibody, but it's all we had. And I thought the biomarker was so promising. We'd already validated it in human tissues, in animal models. We said, this is a great target. And it was checking off all of our wish list, all the items in our wish list. So in any case, we labeled this antibody that was very specific for TREM1. Emily Johnson in my lab helped optimize this. We just did a simple DOTA conjugation and labeled with copper-64, which has a half-life of 12.7 hours. So it gives you a nice window of time to look at an antibody binding and clearing effectively in animal models. And so here's a summary of what we did in an EAE model, which is a model of multiple sclerosis. First of all, we just showed with flow cytometry that TREM1 is really only expressed on the peripheral myeloid cells, not really the microglia. So it's really just able to pass out those peripheral cells. And these are the different stages of that disease. So it's tracking with kind of disease progression, not expressed on a TREM1 knockout mouse. That's our control. And these are some of the images that we have acquired, where you can see in normal healthy mice, you don't really see much outside what's in the blood pool, which is just the heart and descending aorta here. So you don't see much else, which is good. As soon as you have a mouse with a disease, this is before any symptoms, we see it in the spleen, in the spinal cord, bone marrow, everywhere where you would expect some kind of immune response to be occurring. And that signal only goes up with disease severity. So you see the spleen blowing out, and spinal cord signal blowing up, really clear images. This made me so happy to see. We also showed that TREM1 PET is more sensitive than TSPO PET, sort of the gold standard that I just talked about. So our signal to background was higher for TREM1 in every single stage of disease, which is great. And we showed that it's a promising therapeutic target by modulating it with a decoy receptor peptide, as well as looking at the knockout mice, which showed that we delayed onset of disease or diminished the severity. So very cool target. This might be of interest to this group where we're looking at a stroke model. And we just wanted to see, can we detect TREM1 positive myeloid cells in and around the infarct of an MCAO mouse model of stroke? And we could. These are just some autoradiography images. This is a coronal section of the brain. We showed that yes, there's this red signal, high binding of the TREM1 PET tracer in and around the infarct, which makes sense. What we didn't expect, and this is published in Nature Immunology in collaboration with the Andreasen lab, is that we started to see signal not only in the spleen sort of going up, which I did expect because there's going to be more immune cells there after stroke, but we kept seeing signal in and around the gut, in the intestines, that was not there in the sham, was not there in the isotype control injected mice. And that made me think, this has to be real. And I talked to my collaborator, and we took out the intestines, and her group did all the flow cytometry and found that, yes, there's actually TREM1 expressing macrophages in there. And this is something that's kind of seen after stroke. There's this acute activation of all the sympathetic inputs to the intestine that occurs the initial hours after cerebral ischemia that promotes increase in the gut permeability and the subsequent translocation of bacteria, microbial components across the intestinal barrier that contribute to post-stroke bacterial infections. And what we showed is like we could treat with propranolol and sort of close up that gut barrier, and we had better outcomes in the mice. Less TREM1 positive cells were able to get out of the gut and cross into the brain. So it's nice that we sort of were able to look at this whole body immune response in stroke, whereas most people are looking at the brain. We're able to just look at the whole system and understand more about the gut-brain axis and just other components of this disease, because obviously we don't need to take a reductionist approach when we're using PET imaging. We can look at the whole system. Here's another example of using this tracer, but this time in a Parkinson's-like rodent model. This is one of my postdocs, Katie Luco, ran this in collaboration with Tom Montine's lab at Stanford, who's chair of pathology. And we were just looking at 6-hydroxydopamine, which is obviously a neurotoxin that selectively diminishes or damages your dopaminergic neurons. And so she did the stereotaxic surgery there, injected that, and we had a group of mice that was injected just with saline, and we did imaging just to see, can we visualize the inflammation that occurs in a Parkinson's-like rodent model. And again, we were able to do that, but what was interesting is we really saw most of the uptake or increased binding at day 7 after the toxin was injected, and a little bit day 14, but really not significant. So it seems to be an early marker in this kind of a model where there's dopaminergic cell loss. Obviously, this model does not recapitulate all the aspects of Parkinson's, so we're looking at other models. And we're also starting to image Parkinson's patients with TSGO PET. We're hoping to do it with TREM1 PET once we translate that tracer. And the last thing I wanted to show you with TREM1 PET is that we looked at whether we could detect innate immune activation in LPS-induced sepsis. So here where, LPS is a major component of the outer membrane of gram-negative bacteria, we injected that IP, and that leads to a big immune response where you'd see a huge expansion of all your innate immune cells in the lungs, in the liver, in the spleen, and ultimately there's also neuroinflammation that occurs. So we administered LPS 5 mg per kg and then imaged a day later. And this is just showing you our setup where we're imaging 4 mice at a time. They're all anesthetized and they're in this PET-CT scanner. And this is an example of just a normal mouse. And so you can see it's just in the blood pool. It's not binding to anything else. It's just in the heart and descending aorta. And then in a mouse that's been given LPS, you can see there's a lot more signal in the spleen and in the liver and the lungs compared to this vehicle injected mouse. So you can actually visualize TREM1 positive myeloid cells being upregulated and being mobilized and expanded in number, which is, I think, pretty incredible. And then we looked at an isotype control, it was an antibody that would not be binding to TREM1, just to make sure that it wasn't just something non-specific in an LPS mouse. And you can see this looks a lot like the vehicle. So that really proved that we were looking specifically at TREM1 in this image. And then we did the ultimate control, which is looking at LPS injected into TREM1 knockout mice. And again, we didn't see any of the signal that we saw in the LPS wild type. So again, proving specificity. But I know we've got about 15 minutes, so I'll share a little bit about imaging B cells in MS, and then hopefully have a little bit of question time. But just a little bit about this story, something that I became very interested in, really looking at B cells in MS, just after hearing about the dramatic response that a subset of multiple sclerosis patients had to anti-B cell therapies. And I think initially it was like by accident that someone with MS was taking rituximab because they had lymphoma or another condition at the same time. So they were taking that, but they had an amazing remission from their MS. And then they started trials and they saw that, yeah, a subset of patients really responded well, which was exciting, but not all patients. And why that is because MS is really a heterogeneous disease. And it really involves the whole body, your spleen, immune cells in the bone marrow, everything trafficking into the brain, and it can go, both ways. But there are B cell follicle-like structures that are found in the brain of MS patients. And the interesting thing is we don't know this in living people that are walking around, obviously, but upon postmortem, we can see that some patients have a few of these B cell follicle-like structures that are causing trouble. Some people will have 10 to 20, other people would have hundreds of these all over their brain. The meninges in the subarachnoid space is the main part of the brain where you'll find these. And that's why we think that people that have more of a B cell driven disease are the ones that respond. But we don't know who those patients are in order to put them on this therapy more quickly. So our idea was just to label rituximab to start with, and later on I'll show you we've started to look at other traces, but we started with labeling rituximab so that we can inject this into patients, and just with the idea that hopefully this could tell us about, if we were able to give this to people, they could give us an idea from a whole body and brain scan who has a low B cell burden versus maybe somebody else has a really high B cell burden, and we could get them onto the right therapy. And so this is just showing some of the initial results with rituximab using transgenic mice that have humanized CD20 on all of the B cells. And we can see these images aren't as pretty as the TREM1 images. That's because there just aren't as many B cells as they are myeloid cells. So the signal isn't as strong, but we can detect this sort of speckled red dots down the spinal cord. These are B cells. And you see in the normal mouse, you don't visualize that kind of a signal because there are no B cells normally in a healthy central nervous system. And this is just to show you, we take the spinal cord out afterwards, and you can see after removing all the blood that this is the specific binding of this tracer in the spinal cord. So very nice confirmation. And this I really love because we show here that we're getting these B cell follicle-like structures. This is B220 immunohistochemistry. We can see that in the meninges of the mouse brain, really recapitulating what we observe in the clinical condition. And then, imaging CD20 is one thing, but I started to think about the fact that CD20 is only expressed on certain B cells, and we're missing some. We're missing plasmablasts, we're missing plasma cells, we're missing some of the earlier sort of pre-B cells, pro-B cells. And really these plasmablasts and plasma cells are thought to be the key sort of instigators of all sorts of neuronal damage, damage to your CNS in MS patients. So it was sad to me that we were missing them. And also the fact that people started to develop CD19 therapies so that they could target these cells. Thought, well, why don't we make a CD19 PET tracer so we could visualize more of the B cell pool? And that's exactly what we did. And we developed the first PET tracer for looking at CD19 positive B cells, and just using, again, a very specific antibody for CD19 and labeling with copper-64. These are some of the initial PET images. You can see, again, there's not a lot of B cells in the brain, but you can see them. And this is the autoradiography showing that you can visualize the B cells in the cerebellum, also in a lot of the meninges and ventricles. You can see lining the brainstem here. And again, correlating with B cell staining. Mark Stevens and Haley Cropper in my lab did this really beautiful work that's published now in Journal of Neuroinflammation. But I think what's even more exciting is after doing that proof of concept work, Carmen in my lab is making the first human CD19 PET tracer, and she has done that. And this is in collaboration with Viela Bio. They've made a really great anti-CD19 therapy for treating diseases like MS and NMO. And in any case, we thought about using their therapeutic antibody, but then we didn't want to deplete B cells or affect the biology we wanted to study as we were using the PET tracer. And that can definitely happen. We saw that with rituximab. When we used rituximab as a PET tracer, it was actually killing B cells at the same time. So we actually did something a little different, and we used the control antibody that Viola Bio developed, so this mutated variant of their drug, and it's mutated in the way that it lacks the ability to elicit B cell depletion via ADCC due to mutations in the FcR gamma binding site, while it still maintains its affinity for CD19. So it's a great, I think, tool for imaging. We've optimized the chemistry, we've done some imaging, and actually now we're making a fab for this as well that Carmen will hopefully present at WMIC. And this is just showing you some of the results where we can see increased uptake in the spinal cord. We see the opposite in the peripheral pools or tissues, where we see more B cells in the naives than the EAE, which is sort of indicative of trafficking of these cells out of the spleen, out of the bone marrow, and potentially into the central nervous system. So cool that we're learning about what these cells are doing in real time, and we're really hoping to do that in people. And actually we have a grant from the NIH to do exactly that, where we're making sort of a toolbox of different tracers to look at various aspects of B cell responses in MS, so that we can then pick the best one to put into the clinic. So I'm very excited about that. And I think there's a lot of potential because we have drugs that can really help people. We just don't know which drugs to give to which people. And I think imaging can play a big role there. And so just in summary, to wrap up, I have a few points that I'd like you to take away from today's talk. First is that TSPO-PET, it's the most widely looked at and studied sort of biomarker and imaging approach for looking at inflammation. It's been able to give us a number of insights about spatiotemporal dynamics of innate immune activation in different diseases, both in rodents and patients. TREM1 PET that we've developed in my lab appears to be more sensitive and specific for looking at innate immune activation, in particular peripheral myeloid cells. And it seems to be demonstrating utility in stroke, Parkinson's, MS, sepsis. So we're excited about that. And we're actually, I've actually spun out a little company now where we're trying to develop therapies for TREM1 and an imaging agent to be used in the clinic. So I won't be able to continue that work in my lab. We can only work preclinically, but it's exciting that potentially that's going to progress into the clinic. And imaging B cells, I think, has a lot of potential for helping us to select MS patients to receive these promising therapies. So our ongoing work is we're going to be doing a lot more in the chronic fatigue syndrome area, looking at the whole body and brain. We're going to be translating, hopefully, one B cell PET tracer, and we're going to be doing some therapy monitoring studies in rodent models with TREM1 PET. And we've started that, and you'll probably see some of that work being presented at WMIC, which is the World Molecular Imaging Congress. I do want to plug that. Everyone who's in imaging, definitely submit an abstract for that. And I just wanted to say thanks to the team. This is a massive team effort. Anyone who's in imaging and radiochemistry knows that it takes a whole big group of people to do any of these studies. So just want to say thank you. And also thanks for your attention.
Wow, thank you so much. This is just unbelievable in terms of the disciplines that you actually put together. You know, this is basic immunology and, you know, sort of models of disease, radioligand, you know, sort of chemistry, and then translation to patients, including, you know, volunteering yourself, you know, to, to support science. This is unbelievable. Doing this, and so this is a real tour de force. Yeah, so thank you so much, and I'm glad that you're doing this. You know, because it's really not easy to do, right? You have to bring so many disciplines together to actually make this successful. But of course, really great illustration of the power of these tools. And just a couple of background to this is that, even though we're Heart Institute, one of the major areas of focus is actually brain and heart interaction. So this is actually perfect. And yeah, so both Adam and also Ben Rothstein, they actually develop tracers for both organ diseases, and of course, we actually do a lot of work on the immune biomarker aspect, for this purpose. So this is really perfect, and of course. Yeah, and of course, post-COVID, the chronic fatigue syndrome is more topical than ever. Maybe I can start with a couple questions, but I want folks, to really put forward questions or, raise your hand to ask questions directly. So one thing which we are always mindful of that is that, the blood-brain barrier can be sometimes a challenge. And I think you mentioned that in your talk. And it's really interesting to see that some of these markers do go across quite easily. And of course, there are others that are challenging. And you mentioned there may be some ways to predict, as to what's the, the secret passage that one can get across from one side to the other. And also the other question is, like in a setting where inflammation, seem to be more active, does that, sort of make it easier, across the blood-brain barrier? So just your thoughts on that. Very
yeah. So I think it is the biggest challenge for radiochemists is to make molecules to cross the blood-brain barrier if you want to image something in the central nervous system. And so I think for small molecules, there's a number of rules that we try to follow, and it doesn't always work out. We try to have certain lipophilicity and molecular weights and charges, and we follow that, but it doesn't always work, right? And so I have one of my students and I are working on a tool to predict that, sort of an improved version of this multi-parameter optimization prediction tool. So we're using like huge data sets, and hopefully that work will come out soon. My dream is to have an app where we can just take a photo of a molecule and it tells you the likelihood that it will cross the blood-brain barrier. So that's something we're trying to do. But in terms of, you notice that we were using antibodies, like, well, how do they get across? That's like not really a common thing. And I don't think it is. Like, I mean, antibodies don't cross the blood-brain barrier normally. They just don't. They're too big. And I think most things do not cross if they're bigger than a small molecule, and not all small molecules cross. But if there is active inflammation occurring and you're getting trafficking of cells, if you're binding to the cells in the periphery, you can watch that happening in real time. So I think that's a big thing. So there needs to be some kind of inflammatory process occurring. Otherwise, you're not going to see that. But we're certainly seeing it in the various models. And some models have sort of BBB dysfunction. So people will say, oh, well, isn't it just getting across because the blood-brain barrier is all, sort of impaired? But no, I think it has a lot to do with these specific processes that are upregulated. And I could talk more about that, but I don't want to take too much time.
Yeah. No, that's a very good point. Yeah, because we know that inflammatory cells, in fact, have ways of crossing these. So the fact that you're able to tag those, I think is a real advantage. So let me see. So there's one question that what regions of the brain are affected in CFS? Any thoughts? Because you show some maybe even sex differences. So what are your thoughts?
Yeah. So I mean, there are many brain regions that are affected. We still don't really understand it, obviously. But for inflammation, at least, looking at postmortem or looking at some of the results that we're seeing, and also functional MRI, it seems that basal ganglia is very much involved, thalamus, parts of the cortex, hippocampus, because there's like memory issues. So it's a variety of brain regions, and we're still trying to figure out, because there's not a lot of data, right? There's not really a lot of brain banked data for looking at the histology of what's going on. So we're working on building that up a little bit, but that's a really important question, because you do have to kind of pick your regions a priori before you go about your analysis, but it's also such an unknown entity.
Yeah, and the beauty of using these type of PET imaging, I think, well illustrated by a number of your examples. Particularly, for example, the last one on B cell therapies is the ability to deliver precision medicine, right? Because unfortunately, a lot of chronic diseases we deal with are heterogeneous, and that's why finding therapy is so difficult. And so I think the ability to actually identify the pathophysiology is critical. Great illustrations on that. And another question is, from me is that I'm fascinated by the copper labeling, on the antibodies, and I guess I'm trying to figure out in terms of the imaging, especially when you actually try to do, like a whole body imaging. What is the I guess the timing dynamics, on that, so like when is actually the best time to do that, taking advantage of the tracer dynamics as well as the antibody as well.
Right. Yeah, that's important to consider. And it's hard to know until you really try it out in patients. In animals, I mean, I could say probably 24 hours after is going to be one of the best time points, 24 to 48 hours. So that I guess the disadvantage is you'd have to ask patients to come back. So that's why everyone prefers F18 as same day imaging. And so that's why we're starting to look at smaller fragments of these antibodies, but then you trade off on binding. But certainly in the animals, it's anywhere between 12 to 48 hours afterwards. And I think Ben has a question too. I can see his hand. Yeah.
Okay. Yeah. Ben, do you want to just post the question directly? Yeah. So we'll unmute you. Yeah.
Yep. Thanks. Thanks very much, Michelle. Thanks for a very impressive talk as always. Peter stole my first question about how you're getting the antibodies across the blood-brain barrier, so I'll go on to the other one. You mentioned that selectivity of targets is important for interpreting what you're imaging. And specifically, you showed that TSPO is expressed on a number of different cell types. So I wonder if kind of that's your strategy going forward to find more selective markers for different immune cells. Is that the way the field should go? And then how do we interpret, I mean, there's such a wealth of data with TSPO radio tracers. How do we go about interpreting that in terms of what the meaning is?
Yes, really critical to think about. So I'm glad you raised that. It's like what I spend most of my day focusing on when I wake up in the morning thinking about is how to I think the field needs to get more specific. We need to start at the beginning and say, okay, what is the question you want to answer? What are you interested in achieving what's the clinical unmet need? What questions do we want to ask in these different contexts? Spend time with clinicians. And I think what we need to do is start with their data sets. A lot of the times when I've been collaborating with clinicians and people in neurology, for instance, they'll have data sets that I can mine for imaging targets because they'll have human data where they're looking at maybe therapeutic targets or just understanding different processes at a more mechanistic level. They'll have human tissue, human cells, or maybe different animal models. And I think it's important to mine that data or generate that data yourself in collaboration with others because it's a lot of work. And to start at the very beginning, before you touch any chemistry, I think we need to make sure we're going after the right targets. And I think targets that are more specific for cell types, and not just the type, but that tell us about the functional behavior of those cells is critical. We don't have really any good metabolic imaging agents looking at immune cells, I'd say, yet specifically. I think we need those. That's a big unmet need, something I'd like to look more at. But right now we have, and hopefully in the next year or two, you'll see a lot more tracers coming out of my lab because we've spent a lot of time on the front end looking at biomarkers. We have a number of good biomarkers now, and my chemists are busy trying to make these complicated molecules now just to go after these new targets that people haven't made tracers for yet. So we're looking at, ideally, I'd love to have a tracer for every different cell type. And ultimately, it'd be nice to have multicolor PET. That's something that some of my co-workers are developing right now, where you could look at multiple markers at the same time. Because I think these diseases are very complex and it's not adequate just to look at one biomarker. But how do we interpret TSPO PET images? I think it's really important not to draw massive conclusions or sort of these sweeping statements from images. So you saw me just describing some of the chronic fatigue syndrome images now. I can't really say that that's microglial activation. I can't say it's astrogliosis. I can't tell you what it is exactly. I just know there's more TSPO binding occurring. And what we need to do is ultimately compare it to histology. And that's hard because especially in these sorts of patients, it's not like they're going to, they're not going to pass away anytime soon, hopefully. But when they do, it's important to try to correlate that with histology. And I know we're doing a lot of that with Alzheimer's tissue, with Parkinson's tissue. But what you can do when you do have more tissue available for a certain disease is get that tissue ahead of time, stain it with TSPO first, counter-stain it with lots of other markers, and try to understand ahead of time, what is this TSPO, like what are the main TSPO positive cells in this context? And what is the image going to tell me? So I think that needs to be done in order for us to be able to draw meaningful conclusions. Does that help?
Yeah, that's excellent. I think that's the right strategy. Absolutely. Thanks very much.
Yeah, you're welcome, Ben.
Yeah, so that's fantastic. I think in view of the time, so regretfully we have to wrap up this session. Just like to thank Dr. Michelle James for this fantastic lecture, really, bringing us up to date in terms of this amazing field, which is absolutely critical, I think, in moving forward, both in terms of serving our patients, but also, advancing our understanding of disease and devising novel diagnostics and therapeutics. So this is really exciting, and thank you so much for taking time, inspiring, our students, and hope to work with you closely, as we go forward in this area, and particularly, on the brain-heart connection. So on behalf of the entire community, thank Dr. Michelle James for joining us today for a fantastic lecture, and look forward to seeing everybody on next week's program. Take care.
Thank you so much, Eva. Thank you, everyone, for inviting me.